All right, good afternoon, everyone. So my name is Clint Nesbitt, and I'm the Global Director of Regulatory and External Affairs with Genus. You've already heard two excellent talks from our folks and others related to the PRRS-resistant pig projects, so I'm going to skip through some of the background. You heard from Elena Rice earlier in the week to talk about Genus as a company and to give you a briefing on some of the background genomic sequencing that we've been doing on some of our in-house genetics. And also Christine Tate-Burkhart gave an excellent introduction to the PRRS disease as an economically significant respiratory disease in pigs, and some of the cool genetics and molecular biology that have come out in the last couple of years that demonstrate how we can confer resistance to the disease in pigs. So I'm going to skip through that background and instead focus my remarks more on how we're thinking about being able to deploy this resistance trait in a global commercial scale, and then sort of the implications of that strategy to how we're thinking about regulatory globally. So here's the background recap. PRRS bad, gene good. Okay, now you're caught up. Okay, so basically what we're doing to deploy the trait is that we're using a CRISPR-Cas9 system to make roughly a few hundred nucleotide deletion from the CD163 gene in pigs. And as Christine presented earlier in the week, by deleting that sort of recognition domain, the PRRS virus is unable to bind to that particular binding site in the protein on the surface of the mesophyll cells, the macrophage cells, and then that then confers complete resistance in the animals. So what we're doing, and now that we've developed sort of a commercial allele with that particular knockout, the challenge is then how do you get that one allele into all of our elite lines? Two things from a regulatory perspective that I want to point out at this point. Whoops, wrong direction. The first is that because we're using this CRISPR-Cas9 system and all we're doing is making a deletion, the pigs are completely transgenic. There are no inserted novel sequences. It really is just purely a knockout. We're not using templated DNA repair. So it really is truly just merely the deletion itself. Secondly, the other thing that I think is even more important to understand is that in order to deploy this trait at commercial scale, we really can't just make a single edit in one allele and then breed that gene into all of our elite lines. It just takes too much time. It's a lot of breeding. It causes genetic bottlenecks if we were to try to do it that way. It's just impossible. And it also sort of undermines one of the main advantages of the CRISPR system, and that is the ability to reliably make the same edit over and over again. So what we're doing to really scale up this particular trait is that we're making this exact same edit in multiple individuals across 4 of our elite lines. So we're not really starting with one pig and breeding it out. We're literally starting with hundreds. So when you think about that from a regulatory perspective, essentially what we're doing to communicate to regulators is demonstrating, one, we have the ability to reliably make this same edit over and over again, and that the data that we submit about the phenotype and the like is really sort of a representative sample of how the gene works in the animals, but isn't really the way you would think of that sort of the old school event by event regulation where you're providing data about the progeny of every single individual edit. There we go. Okay, so just to kind of scale out a little bit and help you understand how this moves into the breeding population from start to finish. We begin here at the top with what we call the E0 generation, and that's where we're using microinjection techniques into young embryos, zygotes, to really make the first edit. So all we're injecting is the CRISPR and the RNA template. These animals, of course, will be chimeric. But nonetheless, at that first generation, we can still begin doing screening to ensure that the intended edit is present. We can also screen for unintended edits at off targets as well. But the important thing to remember is that we are making literally hundreds of individual edited pig lines at that very first stage and select among those to propagate then down to the second generation. Second generation, which we call the E1, is basically generated by crossing some of those first lines in the E0 to a wild type pig, so that second generation now is heterozygous. And that is where we're starting to really increase the number of pigs for sort of breeding increase and commercial purposes, but we're also continuing to do some genetic screening at that level as well. continuing to confirm that it has the right edit, and that we are paying attention to whether or not there are any off-target edits and the like. And then finally, the third generation is basically where we are now. That's the first time where you have homozygosity in the gene. It's a recessive trait, so you've got to have both copies there to do phenotypic testing. And this is basically the generation where we're doing all of our testing for regulatory purposes. Does the gene work? Does it confer resistance and the like? So what does all this mean for regulatory? Well, to date, we have been focusing our regulatory strategy primarily on FDA. We are sort of midway through that process. We've made some initial submissions to the agency. There are still a few more to go. And the ones that are remaining are basically the results of the studies that we're conducting right now. So the plan is, as they're finished, we'll be submitting those to the agency, and we expect those to be submitted at some point, hopefully in early 2023. The way that the timing of FDA review goes, we're expecting approval from FDA roughly late 2023, 2024, if everything goes well. But we also have to start thinking about our global regulatory submissions. So what we're thinking now that we're moving on from thinking about FDA is where else are we going to seek approvals? And we've made sort of this first cut list now of countries that we intend to sort of target to begin with, based upon the size of the market for the pigs themselves, based upon the likelihood of pork imports from the United States to those countries, thinking about global trade once these are approved, and other sort of key countries in various geographies so that we can sort of get the ball rolling. If your country's not on this list, it doesn't mean that we're ignoring you. It just means that you're not sort of in the first cut of countries that we're going to sort of focus on initially. I'll also mention too that we have a partnership with a company in China that will be helping us to do the regulatory approvals and commercialization in China. And it's also important to point out that for all of these countries, in addition to the United States, we won't be seeking just import approvals for the food. We'll be looking for all food, feed, and production approvals at the same time. So last thought, I hear the buzzer. I just want to kind of wrap up with this picture to help you understand that as we're thinking about our regulatory strategies, getting green lights in all or some subset of these countries doesn't necessarily mean we have that green light to start commercializing, because we have to be very mindful of that really complicated international trade in pigs and in pork products. And we'll have to make some challenging choices about when we feel like we have the right combination of countries to be able to actually start launching commercially. So thank you, and I look forward to the discussion.